Well, in 1998, there were 2 labs working on the phototropins. In 2004, there were over 40 labs. And one of the reasons for that, of course, is that we had turned up some brand new photochemistry. And universities are populated with biophysicists who have very sophisticated spectrophotometers. And they were studying retinal, retinylene proteins, rhodopsin, bacterial rhodopsin, phytochrome. But this was a new one. So they all jumped on it, and all of a sudden we got a huge amount of wonderful biophysical information on the log domains. As I say, there were a whole slew of labs working on them, and there were lots of proteins, different proteins, that have log domains. So I want to finish just quickly by talking about a little bit about these proteins, because this is all very recent. They're distributed all over the place. They're in higher plants, ferns, mosses, algae, and they're in all 3 major groups of fungi, eubacteria, cyanobacteria, even in the archaea. And if you look at them, notice that these are the 2 phototropins. 1 and 2 look like that. Nani has a phototropin. This is sort of a chimeric protein. But then there are these FKF family ones that are involved in day length perception and flowering and circadian rhythms. There's this that's involved in light sensing by neurospora, where it's a transcription factor. This is a transcription factor. I don't know what that is, but then there are love histidine kinases in the bacteria. Some of them have a response regulator hooked onto them, and some of them don't. Here's another transcription factor, and so on. In other words, these log domains are very highly conserved, but the rest of these proteins goes all over the place. Some of them are unbelievably complicated. Look at this one in Halo Orchida. That comes from the Dead Sea, hence Emeritus mortuary, if you know Latin. It's crazy. The past, just goes on forever, and then it comes to a histidine kinase. So a large number of bacteria encode these things. In fact, all kinds of different photoreceptors. Many bacteria have proteins that have just a phytochrome-like protein. Some have just log-domain. This is another chromophore module. Some have 2. Some have even 3. So bacteria are filled with photoreceptors. And some of them are histidine kinases. These are cyclic cyclase phosphatases, and so on. Obviously, helix turn helix transcription factors, and a lot of others that aren't identifiable. And, well, physiological roles. We were able to show that light induces phosphorylation of these histidine kinases. And then we worked with collaborators in Argentina to show that if you kept the brucellosis cells in the dark and did a macrophage assay, they were only 1/10 as virulent as in the light. And if you knock the gene out, the virulence is reduced to the dark level even in the light. So all of a sudden, here is a very important factor in virulence of a very nasty bacteria that causes brucellosis in cattle and undulate fever in man. Well, this is where we are right now. We're in the process of putting together a paper, but we have the sensor protein with its log domain. It phosphorylates itself and then transfers to a response regulator, which then interacts with a couple of flagelllar proteins, and this somehow leads to an increase in virulence. It's also extremely sensitive to light. This is an experiment that Dangshun did, where 10 micromoles per meter squared, which is a very small amount of light, is above threshold already. So it's very sensitive. And now the awkward question is, given this photosensitivity, could enough light penetrate the skin to activate brucellosis? Well, one interesting fact is that if you have undulate fever and are wandering around, you start with your temperature pretty low in the morning, but boy, it just goes up like this during the daytime, then goes down again at night. Well, is that circadian, or is that because you're in the light during the daytime? Is it possible that that could happen? Well, the people in that field are really skeptical. And two of them were in my office just back before Christmas. And we were discussing this, and they were pretty, they were feeling that it was probably really, this wasn't a light sensing thing, it was red ox. I said, well, gee, if it's activated by light, and it only takes between 10 and 100 bacteria to give you an infection, and there's no longer any more light activation because they're deep in the organism, well, maybe that protein that's activated is going to be totally diluted out after the first few revisions, one every 4 hours, 4 to 6 hours. So anyhow, they were skeptical. So it turns out that brucella does go into these monocytes. The monocytes will develop into macrophages, but these circulate around in the blood, so they're actually going through the capillaries. And then when they become macrophages, they get embedded in the underlying tissue. So it could be that they're close enough to the surface to get kicked off and activated. Well, they didn't. The argument was that the blue light just doesn't penetrate. Red light penetrates. But if you put a flashlight up to your hand, you see the red light coming through, but not the blue light. That's lost on scattering. So I finally got irritated. I went downstairs and found a single blue emitting diode, light emitting diode, and this is the skeptics that we have. And there's the light emitting diode, and this is my hand here. That's the finger, that's thumb. And it's that little flap of skin right in there. So now if we shift it over and look at the light that's coming through, I contend that that is blue light going through approximately 3 millimeters of tissue. I should also point out that I didn't Photoshop this. Well, okay, that's where we are. I wandered all over the place. I went back and forth through different eras, if you will. And I had a few fiascos. Took papers out of the notes and so what the heck. I also did a few other things while I was in college. I didn't dare tell my advisors, in music particularly, all the stuff I was doing. They would have realized that that might have come from my crummy grades in the music courses. But anyhow, it ain't over yet because there's a ton of bacterial physiology to look at. So thanks for listening, and I guess that's it. Thank you very much.